Hi, everyone. Welcome to Foresight's Biotech and Health Extension Seminar Group, sponsored by 100 Plus Capital. Really happy to see so many of you here. We're nearing the end of this year, which has been a rather wild one. Had a really wonderful workshop earlier this year, and I'll share the report in this group as well, because I think not everyone has seen it yet. We are already getting our kind of like minds mulling on workshops for next year. So if you have specific ones that you want to collaborate on, please let me know. It is that time of the year where you're planning for the entirety of the next year. And so now is a good time. Just want to put that out there. And I think for now, without further ado, I'm really, really happy to have Diana Guallar here. I hope I pronounced this correctly. And she will be discussing epitranscriptomics as novel players in aging and rejuvenation. And just a reminder to everyone here that you can nominate folks to present to this seminar series, which is how a lot of our recent presenters have made their way here. So thank you so much for joining us. It's such an honor to have you here. It's a really fantastic topic that has definitely popped up in this discussion group before. And for now, the stage is yours. I will be in the chat monitoring questions. I'll share a little bit more about your bio and background here for people to read up on. Looking forward to seeing everyone in the discussion as usual. If you want to make sure your question gets asked, feel free to drop it in the chat already. Otherwise, you can also raise hands afterwards. Okay, well, thank you so much, Diana, for joining, and stage is yours.
Okay, so thank you so much, Alison, and whoever nominated me to be here, and all of you for attending my talk. I don't know if you can see my slides right now.
Not yet. No. I think if you hit share.
Sure. Okay, now. Okay, perfect.
Yeah, let's
go. Just point. Okay, so today I'm gonna be talking about epitranscriptomics as novel players in aging and rejuvenation. So, in my lab, we are interested in understanding how the same genome gives rise to different phenotypes. So this happens, as you know, in individuals that share the same genome or in cells from the same individual which have same genome, but both of them can display such different phenotype, function, and morphology, etc. So we previously knew, like historically, that DNA gives rise to RNA, and then it's translated into a protein, which affects the function. This has been shown to be more complicated than this. We have epigenetic modifications, which regulate which genes are transcribed. Then we have RNA modifications or epitranscriptomic modifications, which I'm going to be talking about today. And of course, post-translational modifications. So the epitranscriptome is composed by a wide variety of RNA chemical modifications. We just know the top of the iceberg about them. The number of modifications is increasing like every month. So we have to be really checking the numbers because this changes monthly. Up to now, I believe there are more than 170 different RNA modifications. Some of this is quite old already, have to update it, but some of them are shared by the three kingdoms of life. Some are specific to eukarya. And this shows that the RNA modifications or epitranscriptomic modifications is a very widely used molecular mechanism to regulate cell function and RNA specifically. So what does RNA modifications do? They can do a lot of things, from changing the charge of the RNA molecule, the base pairing potential of it, secondary structure, protein-RNA interactions, etc. So in the end, both the protein coding of the RNA, the splicing, localization, degradation can be modulated by these chemical modifications. So they have been known for already more, so since the 70s or even before, I believe. But it was not until recently, with a lot of advances, such as the improvement of the methods for detecting them at a sequence-specific, in a sequence-specific manner, that the field is really blooming.
One thing
sure.
I'd like to say, sorry to interrupt. We have to always remember that there are backwards pointing arrows all over the place in these diagrams. Like there's feedback everywhere.
Sure. Yeah, sure. Good point. Thank you. So also they are, some of them are reversible. I will discuss about this later on. They are very dynamically regulated upon stress to make a rapid response of the cell. And they help to determine self versus non-self RNAs. So this has helped a lot, which I will discuss at the end of my talk, the development of this RNA-based vaccine technology. So they are virtually in all types of RNAs, although the most abundant ones, ribosomal RNAs and transferrin RNAs, were the ones to be first studied because of the like an easier way of studying it because of their abundance. So, but now we know that most of the RNAs are or can be epitranscriptomically modified. So now aging, why aging? So I'm very interested in aging because, as you know, it's an unavoidable consequence of living. Now I leave a little question mark. It's the greatest risk factor for most human diseases, and it causes the progressive loss of proper organ function and tissue function, right? And here I'm talking about Europe, but worldwide is the same. So there is an aging of the global population. So it's something that concerns everyone. The discovery or the description of several aging hallmarks, such as genomic instability, epigenetic alteration, loss of proteostasis during aging, was key to advance a lot in the field and to focus in discovering many of the molecular underpinnings of the aging process. But virtually nothing or very little, to be more accurate, is known about what is the role of epitranscriptomics in aging. So what we know is that, we're starting to correlate epitranscriptomic modifications with aging hallmarks themselves. So senescence can be regulated by a variety of RNA modifications, telomere attrition also, etc. But what is the direct mechanism or direct implications of these modifications in aging is not so well known. Also, we are starting to correlate or to see the effect on the deregulation of several RNA modifications in age-related diseases. But this is still not a direct link for now. It's true that few studies, and here I list the main ones, are beginning to directly address the role of specific RNA modifications in aging itself in different cellular and animal models. But for now, as I say, there is not much known in this field. So what is really interesting for me and for the field is that many of these RNA modifications, so this is exemplified here by methylation in the position 6 of adenosine. So they are deposited by writer enzymes. They are then decoded by reader proteins, which do whatever effect they need to do in the RNA molecule, and sometimes also can be removed, like in the case of M6A, by eraser proteins. So this is potentially targetable proteins and enzymes that can modulate the epitranscriptome and are very relevant for the clinics. So regarding aging and aging models, aging and more importantly, rejuvenation has been approached historically through different approaches. So from heterochronic parabiosis to later on reprogramming, somatic cell reprogramming. And today I'm going to talk about one of the models that we use in the lab to study both aging and rejuvenation. So somatic cell reprogramming relies on the overexpression in somatic cells, such as, for example, fibroblasts, so young or old, of the Yamanaka factors or combinations similar to them. So basically, they are transcription factors of the pluripotency state, which induce the reversion of this somatic adult phenotype to an embryonic-like cellular phenotype, so to generate an induced pluripotent stem cell, which is identical to an embryonic cell. This cell can be then differentiated and committed to become a somatic cell of interest, potentially all the cells of our body, which then see rejuvenated, their phenotype. So you can start with an aged cell, which after somatic cell reprogramming and differentiation is able to erase most of the aging hallmarks. So it's very similar to an young somatic fibroblast of origin. Also, some studies are now using, and this is becoming more and more interesting. So instead of going all the path through reprogramming and differentiation again, which can be very, very inefficient and also, let's say, dangerous in vivo, to trying to do partial reprogramming to overexpress with different patterns, like different doses, different time points, these reprogramming factors in the adult or old cell, and rejuvenate partially, at least, its phenotype to an earlier and younger phenotype one. So this has been used and is being used widely, but still there are many challenges that make that this technology still has a lot to give us in terms of basic knowledge. And of course, it's not something that we can think, as it is right now, at least, to use in vivo, but it's very, very informative. So this is our game board in my lab. Basically, we work in aging using cells from young and old individuals, mainly mouse, reprogramming, and we also study the biology of pluripotent stem cells. So I'm gonna go very briefly over the first study that they want to comment today, which is regarding our findings that regarding a specific RNA modification, so HM5C, in pluripotent stem cells. So we published a few years ago a study where we found that the epigenetic factor T2, which you may know because it's widely known for its function in hydroxymethylation in DNA, both to help demethylate and activate gene transcription, but also to deposit 5-HMC as an epigenetic mark itself. So what we found is that T2, even if it lacks DNA binding domain itself, it's, of course, we knew it was able to, it was recruited to chromatin, but we discovered for the first time that it was through an RNA binding protein, at least in pluripotent stem cells, called PSPC1. So in absence of PSPC1, we saw a great decrease in the recruitment of T2 to chromatin. And using diverse models and assays in vitro and in vivo, we saw that T2 indeed needed co-transcriptional, so it needed a co-transcription to be active to be able to be recruited to DNA. Importantly, we also found that T2 was able to interact with RNAs through PSPC1. And among them, we found that many of them were coding or were derived from transposable element genes or transposable element sequences, mainly long terminal repeat ones. So what are transposable elements? They are repetitive sequences in our genomes. In humans, they can be even almost like half of our genome, so almost 50% of it, so it's quite a lot. They come from ancestral insertions of, or they are believed to come from ancestral insertions of retroviruses in our genomes, and they were kept there. And there is a lot of interest in them because they can both have damaging potential if they are mobilized in our genomes, because they can disrupt gene expression and cause changes in chromatin, etc., etc. But they are also key for proper gene program coordination, mostly during early development.
What are these? What are the different classes here?
Yeah, so class 1 is mainly retrotransposons. So the intermediates needed for the mobilization of the element is RNA, which is then retrotranscribed. And here we have different classes. So the long terminal containing ones, so ERVL, ERVs, and other ones, copia, gypsy, etc. Then the DIRs and the line, which you may they are more famous because this line one is very related to its activation is very related to aging, for example, and other diseases, and signs, of course, which are also very, very like commonly heard. And then on the other hand, there are class 2, which are DNA transposons, which is the most classical, like DNA jumping around, but it's not so relevant in terms of the proportion in the genome and also the mobilization potential. So they're quite silenced. So in this case, what we saw is that these kind of RNAs, so transposons, long terminal repeat ones, were the ones, that were bound mainly by T2 and PSPC1, and that this binding was necessary to recruit T2 to chromatin to those sites. So what did T2 and PSPC1 do through binding to these elements? So in specific, this element, so Mervel, which is one LTR containing element which is expressed very early in development but should be repressed in ES cells. So what they did is to catalyze the hydroxymethylation of cytosines in RNAs of these elements. And importantly, this caused the destabilization of these elements. I thought I had it, sorry, I thought I had it here. Okay. So they cause the destabilization of these elements, which should not be expressed in embryonic stem cells in vitro, but which are like cyclically expressed in these cells. So this was quite new at the time we published it, because there were very few studies showing that TETs, which were widely known as DNA hydroxymethylases, were also able to act on RNA. And finally, one thing that I want to point out, because it was also one of the first times that this was discovered, and so was that this RNA-mediated recruitment of specifically Mervel elements, which were modified by methylation and recruited this complex for hydroxymethylation and degradation, also brought together epigenetic complexes such as histone deacetylase 1 and 2 to deacetylase and repress the loci from which these elements were arising. So there was somehow a crosstalk between epitranscriptomic and epigenetic regulation in ES cells. So why are transposable elements relevant in general and in aging specifically? So the young epigenome is mostly sequences that need to be silenced and heterochromatinized are okay. But then with age, it has been shown that there is a loss of heterochromatin, and specifically these elements are very enriched in heterochromatin in young cells because they need to be silenced. So this loss of heterochromatin leads to a transposable element induction, mostly. And in fact, there was a paper 2 years ago showing that the re-expression of this transcript, transposable element transcripts, was very, very well correlated with the passing of time, even more than transcriptional changes that occur at the cellular level with age. So they could be.
I'm sorry, is that a specific transposable element, or is that a whole family?
Yeah, so here they studied a lot of different families. Yeah, so line ones, LTR, other Ervel containing transposons. Yeah, these ones are a lot. Yeah.
Very cool.
Yeah. So, and this is very important because this shows that they can be used maybe as biomarkers of aging. So they have a direct connection with aging, but are they cause or consequence of aging? So there was a very pretty study that just came up this year, showing that this specific transposable element, line 1, that I was discussing before, its increase during aging led to the heterochromatin erosion. So loss of heterochromatin due mainly to its interaction and causing reduced function of this H3K9 trimethyl transferase SOF39H1. So they showed both in vitro and in vivo using progeroid mice and progeroid cell lines that just by modulating or by repressing these line 1 transposable elements, they could restore heterochromatin and other aging hallmarks and aging associated molecular marks, such as they could revert partially DNA methylation age, decrease senescence associated secretory phenotypes, and recover partially transcriptome and proteome to a younger state. So just by modulating line 1 expression, meaning that it's not only a correlation, they increase with age, but they can actively contribute to aging. And also they are potential targets for rejuvenation, at least in cells and in progenic models. So I think this was very relevant to the field. And also what is very relevant is that in line with this, interventions for healthy aging, such as caloric restriction, have been shown to decrease and restore, at least partially, the aberrant expression of these elements to younger levels. So this is very interesting. But what does whether epitranscriptomic regulation plays a role in transposable element regulation during aging is not yet known, something we are interested in exploring. So I want to comment briefly on a second story on another RNA modification, so adenosine to inosine modification in the context of reprogramming. So this modification is deposited by ADAR proteins, ADAR1, ADAR2, and we wondered whether A2I editing was important in reprogramming of fibroblasts. We saw that it was only ADAR1 and its ADAR1 mediated A2I editing, which was required in reprogramming. And here I showed just the loss of function experiment, but we also did it with genetic models. And it was catalytic dependent function of ADAR1 required for this generation of young pluripotent stem cells. And specifically, it was in this earlier phase of the mesenchymal to epithelial transition where A2I editing was required. We found that at the molecular level, there were several RNAs which were not edited in absence of ADAR1, and this caused the mislocalization of these RNAs in the cytosol, whereas they should be relocated in membrane-bound organelles, specifically the ER, so they could be translated into membrane proteins. And we also found that in line with what was published in other contexts, A2I editing loss in this model caused a hyperactivation of the innate immune response that we described was observed. We don't yet know why, but it was observed at the earlier stages of reprogramming, but then was shut down. And this was not possible in absence of A2I editing. And finally, this led to the improper regulation of the unfolded protein response, so which participates in proteostasis during IPS generation, so during this rejuvenation of a somatic cell, and with a final result that these cells could not be generated. These cells were kept at the mesenchymal fate, and we are still wondering and trying to tackle the question of whether epitranscriptomics play a role in proteostasis, not only in the context of reprogramming, but also during aging and rejuvenation of old cells. And so for now, I've shown that I have several aging hallmarks that we have correlated with RNA modification regulation. And finally, we have a challenge that we hope we can tackle, and we hope it's a medium long-term challenge. But what about if there is an altered epitranscriptome with aging? Can we rejuvenate cells by introducing specific epitranscriptomically modified RNAs? So we all know now that RNA vaccines are now, like, we all are getting vaccinated with these messenger RNA vaccines. So in my case, it was Pfizer one. So if you go now to the composition of this vaccine, you see that there was a little word here that is saying nucleoside modified mRNA. So what does this mean? Actually, we're not getting vaccinated with the RNA itself of the encoding for this protein of the virus, but all the uridines were replaced by or were modified by replacing them by an epitranscriptomic modification, which is called methylation at position 1 in pseudouridine. So basically, we are being vaccinated, the ones that got the Pfizer of the Moderna vaccine, with an epitranscriptomically modified RNA. And what is the advantage of using this epitranscriptomically modified RNA instead of this naive RNA molecule? Well, this mark specifically enhances the RNA stability. It increases the protection against RNases. It increases the translational efficiency of this molecule. and also decreases the anti-RNA immune response. And the effect is so big that 2 different vaccines that were being tested, one unmodified didn't go through, and the other one modified had more than double of the efficacy of the vaccine when the RNA was modified versus the one that was not modified, which did not progress. And this technology is being used in other RNA vaccines for Zika, HIV, influenza, Ebola viruses, and this is being tested in different cellular and animal models. And this also has been shown to be important and to be beneficial when you try to reprogram to rejuvenate the cells using RNAs encoding for the Yamanaka factors. If you modify by pseudouridine or methyl-1 pseudouridine, the mRNAs that you use. So using a similar technology, can we now detect and define which RNAs should be epitranscriptomically modified and are not because of the passing of time? And can we then put them back with a proper modification code or pattern and rejuvenate cells? Let's see, we will work towards it. So just to end my talk, I would like to thank the people in my lab, which are working hard, and studying a lab is not easy. So thanks to all of them. And of course, our collaborators and aging funding agencies. Sorry. So, and thank you all for your attention. I'd be happy to take any question you may have.
Wonderful. Thank you so much. This was fantastic. You have a lot of questions already in the chat. I'm going to let you catch your breath for a second. I'm really happy you got to speak to us already. So we have a ton of questions here. So maybe we start out with Abdul Kader and then move to Creon. And that's also a reminder for everyone else here. If you want to ask a question, you can raise your hand now. I'm going to first look through the chat and then we'll go to raised hand. Okay, Abdul Kader, you want to go first? Go for it.
By the way, this is a very interesting topic, and it's really at the very beginning of starting to gain traction. So my first question is really trying to get a similarity here. Many of the RNA are actually catalytic, and they have a cofactor function, maybe with some enzymes. If we take it this way, so enzymes are often changed in their activity using phosphorylation and other modification. Is that the case also? Can we see those changes to the RNA as another way to change the catalytic activity of these proteins? Is that an existing concept?
So what you refer to is RNA modifications which alter their binding to proteins. Am I right? Or
actually, more like if they are altered within the protein structure, like by modifying chemically the nucleotides,
okay.
You could actually change their catalytic behavior.
Okay, so I'm not aware so much of work being done on how epitranscriptomic modifications change the catalytic activity of RNAs that have catalytic functions themselves, and also on whether there is a direct interplay between or cross-regulation between RNA modifications and post-translational modifications. But for sure, this is much more complex than what we know for now. And actually, at the level of RNA modifications being able to change the epigenetic modifications, there is a lot that is being doing now, and so there's a lot of things that we don't know now, and
we have a lot of questions bubbling in. Creon, you go first, and then I'll go to Larry.
Thanks, Alison. Well, I'll ask my second question first, since it's about partially about mitochondria, and that is, do you have anything to say about how autophagy and mitophagy and similar processes, or perhaps more generally, sequences or pulsing or cycling of these processes affects the epitranscriptome? First question. And then my second question is, well, let's do that. I have a quick second question after that.
Okay, so I'm not an expert in mitophagy and autophagy, but yeah, I mean, epitranscriptomic modification is like regulating all the RNA metabolism, no? So really, for sure, it's going to affect everything. And yeah, and I suppose there are studies going the way, but yeah.
In particular, like the question would be, if you're finding these clocks or these markers or these modulators, are they do the autophagy processes modulate the modulators, you know, or show up in changing modulators? Okay, now my first question, which is more of a conceptual one, is if you think about the epitranscriptomal aging or errors that accumulate in a cell as it marches towards senescence, like what's the worst case? I mean, how can I say, which one is worse? A cell that sits there and doesn't do anything for a really long time, like a fibroblast, and it accumulates errors, and then it has to go do something and make something, or make a new cell or whatever. Or what about a rapidly proliferating cell factory that is replicating all the time, which both gives chance for lots of problems, but also error correction mechanisms come into play. So like, is there anything to be gleaned from that conceptually or therapeutically?
Yeah. So the good thing about RNA modifications is that you don't really change the gene so you change the RNA, right? You change the RNA, you modulate the RNA function, but you don't change the gene from which it comes from. So like the information is there intact, and then whatever happens to the RNA can be modulated. So apart from that, I have to say that, for example, M6A modification, so methylation in adenosine, has been shown to be important for hematopoietic stem cells. So many highly proliferating stem cells are also regulated by epitranscriptomic modifications. So for sure, it's going to play a role both in these less proliferative cells, and maybe it's even, philosophically talking, more dangerous if this is altered in some cells that are highly proliferative, as you say, and contribute to such a great number of daughter cells with a wider function. Yeah, so it's very
okay, but the therapeutics might then also be more highly leveraged in such cells. Anyway, thank you for your answer, and that's fascinating talk.
Wonderful. Next one up, Ageless Partners.
Yes, thank you. And thanks, Diana. Very, very interesting. It's quite complex. So really, I just kind of have a question. I'm thinking about like the vaccines, you know, the Pfizer one, as well as these various like gene therapies, in particular, the one that's kind of using plasmids. And sort of have two questions, you know. So first one, is there integration into the genome when that's passed to like additional cells through cellular replication by changing the DNA? And, you know, I'm kind of wondering about that. And also, like, what are your thoughts on the epitranscriptomics within sort of that area?
Yeah, thank you so much for the question. So actually, one advantage of using RNA vaccines compared to DNA, so for example, putting plasmids or DNA fragments, is that RNA itself cannot be integrated into the genome. So it's something that it has also a drawback, which is it's not so stable as DNA. So you have maybe to put it like in several doses, like in more doses than giving the DNA. But it can never be integrated into the original genome because it's RNA, and as long as it doesn't encode for any retrotranscription retrotranscriptase, or is not does not have any sequence which can be retrotranscribed, there is absolutely no chance this will be inserted and be deleterious to the host.
I see. Thank you.
Cool. All right, we have Nicola, and then if we have time, I'll may read a few questions out still.
Thank you. Excellent talk. It was amazing. And I think the nearest field to yours is fusion physics. You have very similar problems that you're solving. You started opening with the fact that you have this huge parameter space that is increasing. there's more and more factors. And my question is about how we should approach the analysis for that. So when we do an experiment to show causality, we try to modify one single parameter and see the effect of that parameter. And that's what you were showing in your results, and it's beautiful. The problem with humans is that we have huge diversity of backgrounds. And so the worms, they tend to not reproduce sexually. They don't like sexual reproduction. So the variability is lower, and it's easier to generate a significant result. In humans, for example, even for genetic variants, we know that the APRI allele is the one that has the most impact in aging. APRI4 is negative for Caucasians, but has actually a completely neutral effect in African descent people. So variability is important on the impact. How this should be studied?
Yeah, so this is a very good point. But the thing, we are like far, like not yet coming to this stage yet. But for sure, this should be explored. One thing that is in line with this is that, as you're saying, so you modify one thing, and there are so many other things that are being modified, right? So how do you tease out? For example, you removed one writer enzyme and SAN2, and there are so many targets, like not only messenger RNAs, but other types of RNAs. So what we are working in is developing, similar to what other labs are doing in other RNA modifications, to developing tools that help us modify specific targets. And where we are using DCAS13, so a similar one to Cas9, but it targets to RNA, and using specific guides that allow to target a DCAS13, which whatever writer or eraser that you want, you can modify specific targets. Because for sure, it's like you remove a writer, you have so many effects. How do you know which one is significant? How do you tease out different effects in different targets? So one approach that we are undertaking to at least try to tackle the relevance of a specific modification in a specific target is using this DCAS13 technology to modify a specific cytosine in this case.
I hear a nod. So then I'm going to take some more time to read out one additional question, and we may get to you, Cosmo, too. Actually, Cosmo, why don't you ask your questions? You can hold them back the entire time.
Hey, so I focus on obesity, specifically the FTO gene, which is the, according to GWAS studies, the most associated gene with obesity. As you know, I see that you published in this area. I'm also focused heavily on this exciting new area called viral epitranscriptomics. And I additionally am really excited about some of the data that's come out over the years showing that there is a very specific adenovirus that is known to cause obesity, not only in humans, but in animals as well. So I'd like to get your take on that. Is there any, do you think there's any truth behind this whole viral epitranscriptomics field that's emerging that suggests that a lot of these modifications are responses or maybe immune defenses against invading viruses, including maybe the retroviruses that are inherent within our genome already?
Yeah, so thank you for your question. Actually, so there is a lot of still technical limitations in the field of epitranscriptomics. So if you rely on very specific mass spectrometry, you have a very good and reliable result, but you don't know the sequence context, depending on how you do it. But usually you don't know the sequence context. If you rely on antibody-based techniques against a specific modification, sometimes there is like, they're not so specific as we would like to say, and to know, to see. If you also rely on bisulfite, the secondary structure of the RNA can also interfere and blah, blah, blah. So the technology is quite limiting yet. So that said, I have just been talking, I was giving a talk 2 weeks ago in Barcelona, and I was just talking about this, about a study that came reporting M6A, like super rich in I don't remember, it was a retrovirus RNA. Once it infects the cell, and then they were at all, like not able at all to reproduce that. And so there is still a lot of controversy and a lot that needs to be done. So I know there is a big hype in the field, and I believe really RNA modifications are very important in the regulation of retroviruses, endogenous retrotransposons too. But I think we need to be careful to really have like a lot of tests, a lot of ways to determine the RNA modification, the presence or the absence, the controls. So yeah, there is some studies that need to be taken with with care.
So we need better tools.
For sure.
We are at time now, and I want to be really mindful of your time. Just as a last question that can be super quick. If people here are excited about your work, how can they help you, your group, move it forward? What are like specific things that people here can do to support you?
Yeah, good question. Maybe trying to find new models where we can apply the results that we are getting in our aging setting. Maybe try to help me because my lab is small, and what we can do is limited. To see if we can validate in other models, in other settings, in other contexts, whether this is universal, whether this is like specific to, in this case, aroblast, or how universal and how relevant these RNA modifications, specifically in this case, methylation cytosines, but this can also be relevant in many other RNA modifications for sure are going to be important there. And so, yeah, I think that this is like the main help we could get to use our system in different, our findings in different models. Yeah. And a lot of help from the chemists, because I need a lot of combinations of modifications in RNA. So yeah, that's
okay, wonderful. Well, we hear you, and we also have the molecular machines group, so maybe that's also something interesting for them we need to work on. I'm going to share again this was the old one, but I'm going to share again the nomination form for future presenters here in the chat in case you would like to propose a presentation. Or in case you'd like to nominate a presentation, that form is here. And I really just want to thank you from the whole group. Thanks a ton for presenting your work. I hope it wasn't the last time that you're here. Thanks for the work that you do. We are rather excited about it, and I'm hoping that that we get to welcome you at some point again in the future. We will have the video probably ready in a few days. And so
thank you so much for having me here, for all the interesting the chat here. Thank you so much.
Thank you. Bye-bye.
Bye.